You co-chair. Gives me great pleasure to co-chair this evening's seminar. Evening for us, Tony and I are both UK based. I'm just going to do a little bit of housekeeping and go through a few aide-memoirs. So we have our Cancer Cachexia conference later this year. Please do submit your abstracts. The registration is now open and the programme is looking excellent. We have our 7th trainee talks coming up as part of the Society Seminar Series. Next one, April the 9th. The call is still open, so please do submit and sign up. That would be wonderful. Do be aware of our mentoring program, specifically for trainee members of the Society. It's an opportunity to discuss your career development, any guidance that might be helpful for you. There's a link here, and I think we'll be putting a link in the chat as well. Also, other opportunities for early career scientists and researchers to increase their visibility on our website and as part of the Society. Please do get in touch. You can share your bio sketches and we can add them. Do join us on Blue Sky or other social media mechanisms. Join the conversation, and we're often active in sharing studies and publications and so on. So please do participate. This is our upcoming seminar on March the 12th. Christian Fraser will be presenting. Please do join for that. But for tonight, it gives me great pleasure to introduce Tony Coll. He's a colleague and a friend. We worked together on one of the Cancer Grand Challenges that we're involved in called CanCan. His talk is entitled Neuroendocrine Mechanisms in Cachexia. He's a clinician scientist. He did his clinical training in diabetes and endocrinology in London, Cambridge and South East England. He's based at the Institute of Metabolic Science in Cambridge, specifically looking at disorders of energy balance, body composition, how this can result from disrupted metabolic pathways, and also in relation to dietary intake, how we metabolize fuel, store nutrients, a lot of interesting and exciting clinical work that's led on the metabolic unit there in Cambridge. As I said, he's an investigator on CanCan. We work together. He's part of the team or looking specifically at how we can develop mechanistically informed treatments for cancer cachexia. And I think I'll stop there and hand over to Tony. I'll just stop sharing, Tony.
Okay.
I'll let you share now.
Can do the same. Can people see that? Is that okay?
It's working for me.
And can people hear me? Is that okay as well? Because can everyone hear me? Can you hear me, Mariam?
I can hear you well.
Okay.
Yes, we can.
Fantastic. Thank you very much. And thank you very much for that very kind introduction. My name's Tony Coll, and I'm based here in Cambridge. And I guess let me see, is that going to work? There we are. Yes. So that is me sitting in my office there, waving out the window on a cold afternoon in February. And you might be wondering why myself, as a physician scientist with a training in diabetes and endocrinology, who works in a building which really was based around trying to understand the causes and consequences of obesity, now finds myself very much enjoying working with a whole range of really great people in the Cancer Cachexia Action Network with Eileen, Marcus, Tobias and Mariam, among many others. And what I thought I'd try and do in the time we have together this evening is perhaps start off with a bit of wider perspective on the sensibilities and approaches that we've taken to try and understand disorders of body composition over the last few decades, talk to you about GDF15, some of its history, and hopefully highlight some of the future exciting prospects which I think are coming through into the clinical arena around GDF15 based therapies. Mention some of the emerging potential targets, in particular some of the work we're doing on MC3R. And again, it's if you like, an old friend with some new investigation as techniques and sensibilities come through, and then give you a little bit of an update on some of the ongoing human studies we've got here, based around trying to understand some of the neurohormonal stuff related to cancer cachexia and eating behavior. So again, taking a bit more of a serious view about why we have focused in on obesity. And the simple message is that obesity harms health. And what we're trying to do in the Institute here is try and investigate the mechanisms through which both metabolic health is maintained, the physiology, if you like, and how this is disturbed in disease, the pathology. And in trying to understand both the physiology and the pathology, I think one of the simple questions, which is easy to ask but harder to understand, is trying to understand the mechanism, the biology behind the variation. This slide that I've shown you here from the UK government tells us what we all know and hear all the time, that a significant proportion of people living in England are overweight, or living with obesity, or living with overweight. But not everybody is. And even within the population, there's a huge amount of variation when people are living in the same environment. And what we're trying to understand is what is the biology which underpins this variation. And at the risk of trying to concertina decades, literally decades of high-end translational research and mouse models, one slide here on leptin deficiency and obesity. I put this up unashamedly because I think it is an extraordinary path that is now over 30 years old, from that initial report of positional cloning of the mouse obese gene and its human homologue back in 1994 to where we are now, where we've actually got some clinical commissioning documents to have this treatment on the NHS for folk who were born with congenital leptin deficiency. Across all of that work, there's been some great work with colleagues that I've worked with over the years, Sadaf and Steve in particular, combining what we've learned from mouse models with extreme phenotypes and going backwards and forwards between various model systems to try and understand not only the physiology, but also the pathology of these systems. And as well as being extraordinarily helpful and beneficial to those who have been deficient in leptin, the whole understanding of the leptin melanocortin pathway, which I'll come back to a bit later, has, I think, among many other things, really put center stage this idea of the brain being very much in primacy of the hormonal control of food intake. And this is a figure from, I think, 2008, if I get it right, yeah, showing that a whole range of different signals, if you like, from below the neck, signal up to the brain in various centers, which I'll talk about in a bit later, get integrated and orchestrated, and then have, if you like, effector arms, which bring about changes not only in behavior, but also particularly how, why, and where you store and metabolize fuel to try and maintain that homeostasis in a changing environment, that allostatic concept, if you like. And you'll see down on the bottom right here that gut hormone, which even back in 2008 was being talked about, GLP-1. And again, I think you can't help but notice that by understanding the physiology of these gut peptides, we've been able to bring about really potent, meaningful changes and delivered to the clinical arena drugs which have, I think, revolutionized our approach to obesity and weight management, bringing challenges, yes, and we're hearing lots about that, but also I think it's important just to really reflect just for a moment just how potent these drugs are. And if you told me 20 years ago that we'd be sitting here being able to prescribe drugs that can pretty safely change about 20-25% of your body weight and really revolutionize people living with obesity, I think I'd be a bit surprised. So I think in all of that journey, if you understand the system, can understand the physiology, you can start to make mechanistically based interventions which can have real efficacy. It takes a bit of time, but I think we have and can get there. To return then to the other extremes, if you like, of that body weight composition arena. And this is a picture I still think is shocking. And I remember being showed this when I was a trainee in diabetes and endocrinology several decades ago. And this is a person living with type 1 diabetes before really advent of insulin. And I was lucky enough to be trained by people who told me right from the beginning that the type 1 diabetes and insulin deficiency isn't just a sugar problem, whatever that means, but it's much more than that. And when you look at somebody with that picture, what you're really seeing is a metabolic and body composition catastrophe caused by an absent messenger. telling you and really reinforcing that issue that if you want to know what a hormone does, find someone who doesn't have it and study them and see what gets better when you give that hormone back. And I think we're all familiar now with people living with type 1 diabetes, but I think this is still a really powerful and stark reminder of the power of hormones to have real impact on your metabolism and your body composition. And again, through my training, I think I was lucky enough also to work with people who had always thought about endocrinology not just as a sort of single molecule, but actually being this widespread orchestration. And we'd always been encountering people who had body composition catastrophes, salt catastrophes, calcium catastrophes caused by rogue messengers, where a cancer cell, for want of a better word, decided it would take on the form and function of a neuroendocrine cell and start making hormones very much out with the usual feedback loop and bring about all sorts of problems within the body, within the person living with that cancer. And I think one of the messages that I want to reinforce through what I'm saying tonight is that I think there are increasing strong signals that metabolic and body composition catastrophes caused by rogue messengers can, at least in part, occur through working and signaling through the brain. And it would be remiss of me not to start talking, I think, at this stage about GDF15. I think it is worth reflecting about where the whole story of GDF15 came from, particularly of where we are now. It hasn't been quick, but I think it has been a really good example of iterative science with crossing over between human populations and mouse models to really understand the physiology and then try and piece that together and make sense of the pathology. I also think it's important just to highlight who I think is the real hero of the GDF15 story, Sam Bright, who's very self-effacing, but very articulate, intelligent, and has done really a lot of the groundwork, continues to do so, about understanding what GDF15 is. It is a member of the transforming growth factor beta superfamily. You can see on the left-hand panel there, it, as so many of its family members are, made with a signal peptide, goes through some maturation and cleavage, and circulates in the system as a mature dimer. Those of you who've been following the literature will be very much aware that when it was first discovered, it was recognized as being a biomarker of diseased state. And it was up in pretty much every disease you could think of that caused harm to humans, be it atherosclerotic disease, heart failure, kidney disease, mitochondrial disease. I show on the right-hand side here some of the human populations and human conditions where GDF15 is really very high, beta thalassemia in particular, and down at the bottom right there, acute infections, which have long been recognized to push up GDF15 to very high circulating levels. Of course, as well as the atherosclerotic disease and renal failure and organ failure, circulating GDF15 has been known to be raised in a range of human malignancies for a long time. And I pick out some of the here: pancreas, breast, colon cancer. Not all cancers, we can talk about that at the end, but certainly circulating levels been raised in a range of human malignancies, and that's been known for a long time. Going back, though, I think to 2007, again, it's Sam Bright who I think has been really pushing through our understanding of this, and a paper that didn't really get the coverage that it did at the time. But I think Sam was really on the money here, talking about some GDF15 producing xenografts in animals that looked to bring about a huge change in the body composition of these animals, which, if you blocked it with a molecule targeting GDF15, seemed to be able to completely reverse a lot of that anorexia and weight loss, which was mediated by what he called MIC1, but GDF15. And again, correlating that with some of the other markers which were hitting the headlines at the time about what is driving weight loss and cancer, it looked like the weight loss in these prostate cancer population really correlated very well with GDF15. And that was back in 2007. Move forward to 2017, another big year for GDF15, where I think a whole series of groups put back-to-back papers out suggesting, and I think still probably strongly suggesting, that this is still where the majority, if not all, of GDF15 action occurs, is in the GFRAL receptor sitting there in the brainstem. We got into this because we were fascinated by that whole idea that you have a circulating hormone which has a very discrete target of action. And this is just a paper that came out in 2019, just before COVID, with us suggesting that GDF15 looked to be a really important part of the endocrine signaling arm of systemic stress. Here, just with some perturbations of some cells, either with a high-fat diet or some lysine-deficient diet, you get an integrated stress response which pushes up your GDF15, acting in the brain, and then bringing about those, if you like, potentially compensatory mechanisms with reduced food intake, aversion to stimuli, and other neural behavioral outputs, which we're beginning to think more and more now about the whole part of the sickness behavior axis, if you like. It wasn't just us that was thinking along these lines. These are some really interesting papers from Tito Borner and Christian Clemenson's group in Denmark, with Tito and his group showing that rather than this being just a reduction in food intake that you might see in a postprandial gut hormone way, there are adverse effects to this. There's anorexia, and if you look in shrews, who are a model of emesis, unlike mice that don't vomit, you can see that this is very much an adverse effect, not just a feeling of satiety and reduction in food intake. Christian Clemenson's group on the right-hand side there also shows that there's a diminished voluntary movement when you give a high dose of GDF15. And although the animals are, if you like, capable of running when they're forced to do so, if you give them a free choice, they volunteer to reduce their activity, very much in keeping with that sickness behavior role of GDF15. And I think in trying to think about this, Steve did a very thoughtful article, if you like, about what does GDF15 actually for. I don't think it was there to have a role in cancer, if you like, but it must have evolved to have some other role outside of malignancy. And I think the best evidence we have so far, there's probably an endocrine signal of nutritional stress, which, yes, is increased with illnesses associated with cachexia, but probably has much more of a role in trying to change behavior to avoid further exposure to that ingestion of infectious agents or poisonous foodstuff, telling you that whatever you're doing at the moment, get the hell out of there, stop eating that, move away from that environment, and bring about that canonical sickness behavior that you see so often in animal species, where you move away from the noxious stimuli, hide away in the corner of your cave, the corner of your bed, and wait for everything to settle down. And part of that sickness behavior made us start thinking a little bit about corticosteroid, which has been well recognized to be part of that acute response to a stress, to an illness. Many of you here will, of course, know as well that corticosteroid has a huge amount of effects all through the body, not least of which on skeletal muscle, because if you're giving it exogenously with big doses of therapeutic glucocorticoid, you get that well-recognized feature of muscle wasting. So with Iris Amino, one of the postdocs in the lab here, we undertook quite a range of studies, which are now published a few years ago, showing quite convincingly, I think, the activation of the HPA axis and a drive both in acute and slightly longer term corticosteroid levels can be brought about by both exogenous and endogenous GDF15. And of course, that made us start thinking, well, if you're putting your corticosteroid up after GDF15, could that actually be impacting upon some of the loss in lean mass that you see not only in acute chronic illness, but also when you give people GDF15 for a long time? This is some unpublished work from a series that Ira has done, giving a long-acting form of GDF15 to some animals, and you see both a reduction in fat mass, but also a quite marked reduction in lean mass. Ira's gone on now to make a muscle-specific model of glucocorticoid just missing within the muscle, and I just show you some evolving data of some markers of muscle atrophy after an acute dose of GDF15. You can see in the wild-type animals given GDF15, you get this significant increased expression in these muscle atrophy markers, which is completely absent in these animals that are missing glucocorticoid receptor just in their muscles. We're going on now to push this further by using prolonged courses of GDF15, looking to cross it with some cell lines which are have malignant potential and are pushing out high levels of GDF15 to see if we can bring about a bit of a model system outside of this acute scenario. But watch this space, and I'll be very happy to report back later on in the year as that story evolves. What about GDF15 and humans then? Of course, you'll be very familiar, I think, with this study from the TRACER-X consortium, which came back in 2023, which showed a significant association between circulating GDF15 and loss of body weight, skeletal muscle, and adipose tissue. And again, made the very good comment supporting that the therapeutic relevance of targeting GDF15 in the management of cancer cachexia. And again, I'm sure you'll all be very familiar with this paper that came back at the end of last year in 2024, using ponsegromab in the treatment of cancer cachexia. Just to remind you, relatively small number of patients, only 180 or so, mean age 67, all had elevated levels of GDF15. A normal healthy person, you'd expect to have a level between about 200 and 500 picograms per mil. And this was a combination of people living with non-small cell lung cancer, pancreas, and colorectal. But as the text on the top shows there, after 12 weeks, they had a significantly greater weight gain than those in the placebo group. I thought this was a really exciting paper and really pleasing to see that work from animal models that had gone on to move into the clinical arena. And I also thought it was very pleasing to see some very encouraging editorials coming through, which were really trying to bring back some of these themes which I talked about that had been sort of bubbling under, if you like, for some years, and bringing back into focus the role of the brain and bringing about what we're seeing in patients living with cancer cachexia. I think in this graphic here from this particular editorial, I think there's a really nice addition here about the chemotherapy aspect here. And I just couldn't help but show this data from Dana Breen, which I think is really interesting and something that I think I'm looking forward to seeing how this translates into some meaningful impact, with GDF15 neutralization being a really potent way to alleviate that platinum-based chemotherapy emesis that you see. And I would recommend looking at that paper if those of you who haven't seen it before. Maybe we can discuss at the end about where we think this is going, but I think there's some really exciting stuff here about how GDF15 might bring some meaningful, GDF15 targeted therapies might bring about some meaningful hard endpoints in patients living with this condition. Let me stick with the brain then and move on back to my old friend, the leptin and central melanocortin pathway within the hypothalamus. You can see on this graph on the this graphic on the side here, this leptin melanocortin pathway, where the leptin from the fat cells moves and targets leptin receptors in two competing first order neurons, one which increases food intake, one which diminishes it, before it then goes up and targets MC4 receptors on second order neurons in the brain. The MC4 receptor, I think, has really been in the spotlight for the last 20 years. It's so famous, it's got its own website. This is something hosted by SADAF, so you can have a look to see what the developments are there, not only in the physiology of this, but also the pharmacology of this, with some targeted agents on that particular receptor. Somewhat in the shadows of its more famous cousin, though, is the MC3 receptor, which we've known about for 25 years. And I think it's very important for me to acknowledge the fantastic work of Dan Marks, Roger Cohn, and Andrew Butler over the last 25 years, and really pushing away at defining the role of the MC3 receptor in a whole range of different mouse models. And of course, Dan and Roger were very much on the money in this paper in 2003, I think, by already highlighting the potential role of this receptor as being a target for cancer cachexia. Over the last 20 years, I think a lot more of this has been focused in on mouse, but we got much more interested, I think, in 2021, where I think it was a little bit of that issue of a question, particularly, which had never been really resolved, at least to my satisfaction, of what the role of MC3 receptor was in the human population. And I think over the last decade in particular, the growth of very highly curated, genetically annotated human populations, such as Genes and Health and Biobank, and the ability to have even more accuracy in phenotyping single cell populations, as well as all the computational power that these human and mouse techniques require, came together with this paper which we published back in 2021, very much linking MC3 receptors in humans to childhood growth and the timing of puberty, and building on a lot of that mouse model work, suggesting quite strongly that MC3 has a key role, not in perhaps driving appetite, but determining then where you dispose of that acquired energy, both in processes like growth, going into puberty, and the development of lean mass. So determining where and how you spend and place your resources that you've acquired within your body. The other thing which I think is really interesting, going back into the mouse models, is this really nice paper that Roger Cohn did in collaboration with Richard Simile. And I just showed two really nice figures here from that particular paper from the Simile lab, which indicates that although the MC3 receptor neurons are at the base of the brain there, they project to a whole range of different regions within the brain that have a role in behavior and volition and reward and motivation, suggesting that the MC3 receptor is not just there to acquire those calories, but has a much wider role in how you move and react to the external environment. And therefore, why focus on MC3R? Well, I think I agree with that commentary that Dan and Roger wrote back in 2003. This looks to be a potential target. And if we can understand what aspects of melanocortin biology are relevant to the phenotypes we're seeing cachexia, can we start thinking about targeting that receptor to bring about some meaningful change in conditions where maintenance of lean mass is a problem? And Roger has already started publishing quite a lot on this, and I've just highlighted one of his papers in Science Translational Medicine about some reagents that we can really start thinking about to bring some changes in body composition in a good way. We've got MC3 knockout animals back in-house, and I just highlight this to say that we're beginning now to undertake a whole series of murine experiments to target and challenge the MC3 receptor to see if we can bring about some of those meaningful changes in lean mass. These are just some largely confirmatory studies about the MC3 deficient phenotype, which I think is really important to remind ourselves that this looks to be a robust phenotype now as much as it was 20 years ago, where you have reduced lean mass and altered response to fasting in the MC3 deficient state. If you look at that top right panel with the wild type mice and energy expenditure, if you fast them, the typical normal response is to reduce your energy expenditure. This doesn't seem to be the case in the MC3 knockout mice, suggesting that they have a real difference in how they partition and move things as they transition from various nutritional states. They also, as has been shown many times before, have altered activity, both in day-by-day living and on treadmills, how far they run, how willing they are to run. And I just highlight some of the evolving animal data that we have within the kit, within our Animal Institute. They also, of course, have altered endocrine axis. And I show this from knockout mice with their IGF-1, which is significantly different compared to the wild type. And in the paper that I mentioned before, that from 2001, we showed some data suggesting that there was a key population of MC3 receptors on the GHRH neurons, and we're going on now to try and delineate that with a series of mouse models to see how much of the MC3 phenotype, how much of the action of that goes down that particular GHRH axis. I mentioned again one of my favorite hormones, corticosteroid, because again there's a change here in the feeding and fasting response that you see between wild type and knockout animals. And we're undertaking a series of experiments now with some isotopes to see if we can work out what it is about the MC3 receptor deficiency, which means that reagents ingested and stored get moved around between the body, between different states in the feeding and fasting transition. I mentioned corticosteroid again because I think that links into one of the previous really nice adventures that I had in metabolism, cancer, and immunology. And this is a graphical abstract, if you like, from a paper that I had the good fortune of being involved with Tobias Janowitz and Doug Fearon when they were both here in Cambridge, suggesting that tumor-induced interleukin-6 makes a real impact on host metabolism, and in doing so has an impact on anti-tumor immunity. Corticosteroid, of course, has a role in metabolism, but it also has a very powerful role in immunology. And in this particular model system, which you can go and read about, we were suggesting that the high levels of interleukin-6 had an impact on the ability of the liver to modulate and respond to caloric deficiency, which then in turn had a role in increasing glucocorticoids, which in their own turn had a role in changing, if you like, the immune microenvironment. I just show one panel from that particular paper showing that if you increase the corticosteroid in these animals, you had a fall off in the intratumoral CD3 count, highlighting once again that hormones such as corticosteroid have pleiotropic actions with so many different systems talking to each other, which can be complex to understand, but I think is the nature of what we're trying to unpick when we're trying to understand conditions such as cachexia. And in trying to come off the back of that story about corticosteroid and metabolism, it was some really helpful conversations with Tobias and Doug and the crew over in CR UK at the time, which led us to set up with Pancreatic Cancer UK and more latterly with the wonderful funding from the Cancer Grand Challenges, the MyPac study, which is exactly what it says here, metabolic and immune phenotyping in patients with cancer. And this has been fantastically led with Claire Connell, who was a clinical fellow and has now recently become part of the staff here in the oncology team here at Addenbrooke's. And this was very much an attempt to bring some of those sensibilities that we'd had in our translational research institute outside of my window here, to study people's body composition, to study their energy expenditure, and also to study some of their responses to some meals that we'd had quite a lot of experience and experience before in trying to define hormonal responses to these nutrient challenges. So an observational study looking at systemic metabolic and immunological changes, looking at people living with cancer, primarily people living with pancreatic cancer, those who are weight stable, those who are losing weight, compared to those healthy volunteers. I would say as well, in particular, that baseline study and moving into the overnight stay, we tried to focus in particular to try and get a better handle on what was really happening with that HPA axis, both by taking salivary cortisol, urinary free cortisol, and also sampling of it while they're overnight. Again, very much work in progress here, but I just wanted to highlight that in looking at some of the energy expenditure in these participants, we're using both double labeled water and resting energy expenditure in indirect calorimetry. And these are some data from our initial preliminary analysis at the tail end of last year, I think, or the year before. What I wanted to say about this was to really highlight the work of Michelle Venables, who had undertaken an awful lot of normative data from the National Diet and Nutritional Study. So the blue dots here are not healthy, young, 21-year-old students, but actually a whole range of age-matched people, which I think gives us a much better normative spread from which to compare our participants. And again, on this very small sample, it's looking like there isn't any increase in energy expenditure, but looking like that energy expenditure, such as we can measure on that preliminary analysis, sits very much as you'd expect for the fat-free mass within these particular individuals. Again, very much work in progress, looking at trying to see if we can get a handle on any novel signals, any novel biomarkers about that will distinguish between those who are weight stable and those who are losing weight. These are some very preliminary data from the peptidomics core, which Richard Kay and Fiona Gribble, my colleagues in the next office here, have been undertaken when we first got some samples coming through with this study. And again, on this very crude principal component analysis, we are seeing a very different pattern in those people who are weight losing versus those who are weight stable. And we very much look forward to seeing, I think, that the full fruition from this study coming back from a whole range of different discovery and peptidomic platforms later on this year. Let me finish then by coming back into the brain, if I may, and highlighting a study that I've had the good fortune of being involved with some colleagues from cognitive neuroscience and psychiatry, in particular Naresh and Paul Fletcher, who have been really making a great deal of progress, I think, in trying to combine cognitive neuroscience with metabolic science, not so much in the arena of cancer cachexia, but very much in trying to understand quite a lot of the appetitive and consumptive behavior in people who've been living with other disorders of body weight and body composition, initially with obesity, more recently with people living with anorexia nervosa and bulimia. Now, I don't for a second think that these conditions are the same, but it felt to us that all of these have disorders of appetite and consumption, and could we use the tools of cognitive neuroscience to try and understand what may be happening in people who are living with cancer and are having adverse effects on their body composition and their appetite. So this CANCOG study is a very simple study. We're looking to get people up to come to our facility just for the morning to undertake some self-report questionnaires, some computer-based tasks, some bloods for some archive analysis later on, and then also some functional MRI work to see if we can use these cognitive neuroscience tools to get a handle on what's happening. And I'm just going to walk you through a little bit of some of the techniques, again, very much work in progress here, and I very much hope we'll be able to report back in the latter half of this year as we get more people coming through. But I think it's important for us to define a little bit about what we're trying to get a handle on here, because appetite means a lot of different things to lots of different people. So how we view this is at least 3 of the different subcomponents of appetite. And these processes, and I haven't got time to get into this today, have been shown to be dissociable and underpinned by different mechanisms in a whole lot of different animal studies and human studies in the past. There's that motivation for food, that basic drive to seek and consume. There's that valuation of food, to the extent to which you value things, and which then underlie the choices you make. And then there's that hedonic, that pleasurable aspect of each of these things. We can argue, and people often do, that these are not indivisible, but they are interlinking. I think that is the nature of appetite. But I think in trying to pull apart each of these, I hope we're trying to phenotype in a little bit more detail than just asking somebody, do you feel hungry? Because there's a whole complexity, I think, about that simple act of eating something or not. So let me just walk you through some of these tasks that we're using. This one is a visual probe to measure motivation. And it tests motivational stimuli in an individual in terms of how invigorated they are in undertaking an action. And the simple concept behind this is that if 2 stimuli are presented together, if a person finds one of them more motivating and therefore more attention-grabbing, they will respond more quickly to a subsequent instruction that appears in a place where that stimuli was previously cited. And if I show you what these people see on the screen, you can laugh or criticize our choice of stimuli. We are trying to measure motivation to some high rewarding food, to some low rewarding food, green tea versus radish salad, against some non-food, cute puppies or traffic cone. And these are some of the examples of the images that we show these people on these computer-based tasks. And again, walking you through this slowly, there's a text down the bottom which tells me what we're doing. But if you look on the right-hand side, you can see what people see when they do the tasks. They sit in front of a computer, they see a prompt in the middle to say fixate on that spot. Then you see 2 pictures appearing side by side. The pictures will disappear, and then a probe, either up or down, will appear behind one of those pictures. And simply by responding on the up, down, left, right cursor panel on the computer, you can ask people to make a signal. If they've been looking at a particular side, they will respond more quickly to what they have previously been seeing. In terms of valuation, that willingness to pay, we do another simple task on the computer where we have an imaginary auction type task in which participants bid for different foods from a limited budget. 1 bid per trial, and at the end, the trial will be chosen. And if they've outbid the computer, they will receive the food. So you have to sort of go with the flow. But the optimal behavior here is to bid what you think something is truly worth. And once again, we can talk and you can critique our choice of foodstuffs here, but we're trying to go through a whole different range of foods that people might say, yes, I would quite value that. I would like to go for that. And you can have 1 image per trial. And once again, you slide very simply on the cursor keys how much you think this is worthwhile, and then you press your bid to see how much you think. And you're kind of bidding against the computer. So if you really want something, you will invest a little bit more into that. Finally, what about measuring reward, the functional fMRI food reward task? Again, participants do this in the scan. It's non-invasive. The task is optimized to activate that brain reward system. And once again, we present people with food, high reward, low reward, and non-food. And we ask people to hold down how much they enjoy, reward, or feel pleasure by looking at these particular images. And the longer you hold down, the longer you indicate your interest in this particular image. We are currently undertaking our first batch of analysis on the first 10 participants living with cancer versus age-matched healthy volunteers. And the planned analysis is to try and compare the food versus non-food for each of these participants. I just show you an image from a study that Paul had done some 10 years ago from a previous study with a similar task, showing ventral striatal activity from high reward food. So very much work in progress, but I think we are very much hoping to get a little bit of an insight into some of this cognitive processing, which we think may give us some clues as to what is happening to people who are living with cancer and have lost their appetite and are living with a reduced body weight. I think I am nearly at my last slide on that one. So my take-home message on this one then is that I think behaviors like appetite must, at least in part, like all behaviors, be mediated through the brain. These hormone pathways, which I'm talking about, which are often at the heart of so many of these behaviors, are complex and multi-layered. And I pick up that hormone corticosterone. It does lots of different things to lots of different fields. But what I think has been very helpful about being involved in the whole Cancer Grand Challenge project is that when these fields meet, when cognitive neuroscience, immunology, cancer biology, metabolism come together, I think you can really start to see if you can work out whether you've got some real targets for meaningful intervention. And again, I think what I've hoped I've demonstrated is by combining human phenotype with mechanistic models in animals and otherwise, you can bring progress in developing effective treatments. It's not quick, but we have got there with metabolism, and I'm really hopeful that we can get there the same way in cancer cachexia. I better just show my last slide, otherwise I'll get into trouble. Yes, the team. I hope I've mentioned some people along the way. I've got to mention Steve and Giles, who have been colleagues in the IMS for more years than I can to mention, for whom I owe a great deal. Ira, who is sitting, she's standing there right at the end there, and Mattia in particular been very much driving some of the animal work. If I've missed people out and if I've misquoted along the way, that's my human fallibility, and I apologize for people watching this on the rerun. But I think I will leave it at there and hand the screen back over to Mariam, and very happy to chat and answer questions.
Thank you so much, Tony.
Shall I stop sharing? If
Yeah. If you stop, there we go. Thank you. What a wonderful talk. I can't.
Have I stopped sharing?
Yes, you have.
Have I?
Yeah.
Can you all see me? You don't have to. I'm sorry that this is what I look like. But...
Well, I can see you. I can't actually see others. I can see the participants named, and I don't see any questions in
okay.
Q&A.
Yeah.
So I don't know, Fabrizio, if you can see any questions that I can't see or hands up, but please.
I can.
You can't either. Please do raise your hand
okay.
or shout out if you have a question, and perhaps I could get us started. Tony, you made me think a little bit about lung oncology and how it is not uncommon to see immunotherapy-related toxicity in our patients, whether it's pneumonitis, skin reaction, or hepatitis, and so on. And often we get these patients back on checkpoint inhibitors by giving them a course of high-dose prednisolone, starting off whether or not they need IV methylpred dosed or just oral. But invariably, they need to stay on a maintenance dose of steroids whilst they go back on checkpoint inhibition. Typically, it'll be around 10 milligrams of prednisolone. And just anecdotally, I'm often seeing that these patients might then tolerate checkpoint inhibition, but they do seem to be on a downward trajectory for weight. And you've kind of made me think that I've never put two and two together and thought a downward trajectory of weight on long-term steroids. But what about in terms of immune therapy response? Because if I understood, IL-6 driven inflammatory response leading to increased corticosteroids, reduced ketogenesis, and then possibly reduced immunity. I've never quite looked at what that impact does in terms of IO response.
Yeah, I think there's a lot that we don't understand. I think there's a lot that where with my pure sort of hormone head on, every time I see dexamethasone, I get very nervous about high blood sugars, muscle wasting, all the side effects that the 20, the bad things that happen with dexamethasone. I mean, I think it's an extraordinarily powerful drug and it has its place. And I think it's a really difficult balancing act. I mean, just talking about that stuff with Tobias and Doug, I think what we were trying to say there is that so much of metabolism, when you press on one thing, you get something else going somewhere else. And it's this sort of trying to play whack-a-mole, trying to work out how do you get that balance right? Because if lots of interleukin-6 is going up and is reducing your appetite, the normal response to a reduced appetite is to increase corticosterone. It's been well recognized that fall off, certainly in fasting, can put your corticosterone up as a sort of a stress response. But then how do you balance all of these things? And I think what I'm learning is more and more that I don't know. And that was really a bit of the impetus to undertake the MyPack study and say, can we really get, rather than have an idea, why don't we just observe what's really happening so we can get something a little bit more robust data to see what is really happening to that phenomenally complicated thing that is the cortisol pattern during the day. Because as an endocrinologist and physician, my answer to everything is it depends, and I don't know, because it really does. It's so variable. And I think we even started talking about at the time, why do we always give medicines in the morning? Maybe because that's when the cort's high. What if we gave medicines in the evening when the cort's lower? Would that be better? And I'm not sure we even know the answer to that yet. So I think there's a whole lot of unanswered questions that I don't think we were even knowing were a question before. Do you know what I mean? When you start having fields coming together. So I don't know.
Yeah. But that's really fascinating because, we know that there's diurnal variation in terms of cancer cell dissemination depending on daytime, evening, and yet we don't use it to guide when we give chemotherapy to our patients. I wonder if that would potentially impact the steroid.
And again, to me, like to a man with a hammer, everything's a nail. I think corticosterone is important. Is it the be all and end all of everything? No, but I think there was enough compelling data that I think everyone agrees that all of these drugs, all of these glucocorticoids, which have potent effects and are really helpful and wonderful, do have downsides as well. And we recognize that. And it may be that perhaps we haven't thought hard enough about what the impacts are on that. But that's all to think about and all to do.
And I've got two questions. So Noor Aziz has asked, are there any studies that connect or relate leptin and GDF-15?
Leptin and GDF-15 connect. I think those two particular hormones have distinct roles biologically. So GDF-15, when you're well, when times are good, it sits along doing not very much. And there are some very recent reports of some humans who are absent of GDF-15 that came out recently, and they seem to do just fine throughout the world. So contrast that to leptin. If you're missing that, you're in all sorts of trouble. So I think they were very different things. I think leptin is one of the critical signals of housekeeping, telling you that if your fat mass has drifted away, you need to eat, you're dangerously low in your stores. I think GDF-15 sits in the background there and it's like a little central hormone. If things are good, thank you very much, you don't need me, but it's there to say things are bad, change it. So I don't think there's much to connect the two of them together. I think they have very distinct roles, and it's certainly in my head. I don't know if people were thinking about a particular question about using it as therapy to change body weight in a particular way. I'm not sure what people are thinking of. Sorry.
And you said distinct a couple of times. One of our hypotheses, and others have said the same, that there are these distinct clinical cachexia subtypes. So when you talk about these potential neuronal cognitive hormonal, that there are different pathways involved, whether it's anhedonia, anorexia, or so on.
Yeah.
Do you expect that if it's just muscle loss or fat loss or a combination of the two, the way we see it in body composition from TraceRx and others have shown this, that there might also be underlying specific neurohormonal pathways that are unique to
Yeah.
the
I,
body
I,
components?
I think we're only just beginning to
Yeah.
understand our ignorance and the way that we always try and put things in boxes and labels, if you like. And I say that from the metabolic point of view, from which is where I'm coming from. Causes of obesity, lots of different ways you can get there. Type 2 diabetes, it's a very much heterogeneous condition with different organs having that communality, if you like, of just having a high glucose. But the way you get to that can be very different. So I don't see why it can't be the same sort of thing in people who have lost their appetite and maybe are losing key components of body composition, if you like. There may be lots of different ways that common phenotype, which we think is all the same, it's not. It's probably coming from this way and that way, and it's trying to move back and getting, you know, the subdivision and trying to get a bit more of a handle on the phenotype rather than just saying, have you lost your appetite and have you lost weight? Getting a bit more of a depth in the phenotype and asking how people are feeling and thinking about their food and their environment.
Well, if I understood the MC, and I've got the luxury of asking you the questions because I'm waiting for more to come through on the Q&A. But if you showed with the MC3R deficiency, there was this impact on lean muscle. So it could very well be that there's something that drives adipose tissue loss, something else that drives muscle loss. I've got another question. What is the effect of decreased GDF15 on the immune response?
Decreased or increased?
Decrease.
Decreased.
Because Tony, we know there was that paper last month, and I knew about
Yeah.
the GDF15 creating an immunosuppressive
Yeah.
ME and how that might impact checkpoint inhibition response. But I
I
don't know
think
about
that's,
decrease.
I don't know about decrease. No, so what were they doing there? They were there was that combination, wasn't it, of using checkpoint inhibitors and also blocking GDF15
Yes,
and
yes.
having what looked like a good impact, a
A better
positive
response.
impact on patient outcome, which is great. And then people start wondering, well, how is that working? And I think I've focused very much on the brainstem, and I still think, at least to my reading of the literature, the receptor of GDF15, I think the evidence about it being in the brainstem as being the only receptor that we know about at the moment, I think is still robust. Even that paper, and I think some of the editorials around it, were saying we're not quite sure whether that effect on the immune system is going through the brain, maybe even through corticosteroid, or is there a to be determined another receptor there. I think I saw a paper on trends in endocrinology metabolism. So the mysterious GDF15 hormone. So there's always more to find out, but I think, you know, if it's making a difference and the concept of having, you know, combination therapies is well rehearsed, and why can't we have 2 and 2 making 5, if you know what I mean, and that synergy between things. But I don't think we've fully bottomed out on that, but I do wonder whether at least some of that impact on the immune system may be at least in part by having an impact on circulating corticosteroid, which is so profound. Even if it's not flattening it completely, what if it's perhaps changing even some of the diurnal variation? And I don't think we know that yet.
No, we certainly don't see in non-small cell lung cancer that if GDF15 is increased in the blood, that the TME is anyway immune suppressed or immune cold. So I think you're right. We don't quite know the mechanism. I get to ask you another question because I'm going to hold on until the hour, unless anyone else wants to ask. What about, sometimes we talk about the complexities of trial endpoints, measures of readout. You talked about the anti-GDF15 monoclonal study looking at stabilization of weight, but also increased weight and some other secondary endpoints. Tell us about MyPack and some of the clinical work you're doing. Any issues with getting ethical approval or having phenotype
So I
that's
think,
feasible?
yeah, in terms of those studies in people living with cancer, where what we were a little bit fearful of is that we were adding a burden to somebody whose life is already complicated. And I'm always amazed at just how wonderful people are to, even when you say to them, we're not offering a CTIM, we're not offering an intervention. This is an observational study. We just need to try and find out what is happening. We might not be able to immediately offer a fix for you, but if we can understand what is going on in your body, then maybe we can, perhaps in the longer term, help work with your clinical team to guide therapies that might make you feel well while they get along and do their really important job of managing and tackling and the cancer itself. We were a bit worried as well that and it came up in ethics, that will these people find this sort of stuff too tiring? And that's where we went for the sort of the Coll thing about modeling it in a morning in clinic, if you like. You come up, and we were trying to do it as simply as we can. A lot of the questionnaires that we did, we've also put onto a remote platform, so they can do it at leisure in their own home. So that was a sort of a refinement to try and say you don't have to sit in a classroom and do an exam. You can do it on a laptop or an iPad at home. And I think the majority of people are coming up, and Claire's not here to talk to it, do manage it okay. And I am forever grateful, and I think I'm impressed by just how wonderfully generous people are with their time in a point in their life where there's a lot going on.
Is the patient altruism that we see in these studies is remarkable.
Ordinary, yeah.
I'm gonna end it there, and I'm gonna thank you on behalf of the Cancer Cachexia Society and all our members, and thank you so much for giving.
Thank you very much for the invitation. It's been wonderful to talk and think and have discussions with people who up until now I haven't had a chance to talk to, and it's been really enriching. And thank you very much for the invitation.
Thank you very much, Tony. Okay, thank you.
Have a good evening, everybody.
Bye-bye, everyone.